Hello, everyone. I'm Shannon Wyman, Director of Program Development at Keystone Symposia, and it's my pleasure to welcome you to today's ePanel. Today, we will be featuring the awardees of the Michelson Prizes Next Generation Grants, an international prize that supports early career researchers who are applying disruptive concepts and inventive processes to advance human immunology, vaccine discovery, and immunotherapy research. The prize is a collaboration between the Michelson Medical Research Foundation and the Human Immunome Project to award $150,000 research grants annually to support young investigators from a wide range of disciplines. Here to share a few remarks about the prize is Dr. Michelson himself, founder and co-chair of Michelson Philanthropies and the Michelson Medical Research Foundation.
Morning. Thank you everyone for joining in. A special thank you to you, Wayne, and the Human Immunome team for all of your hard work that made this possible. But what today is really about are this year's prize winners. Since it is actually a stated purpose of the competition to fund daring or high-risk research, people often ask me how often the research has actually succeeded. Always. The answer is always. In every instance, our prize winners have established themselves as capable scientists, able to independently conduct their own research. They've been able to obtain follow-on research funding to continue on their own work. Some have rapidly gone on to head even broader research efforts in various university settings. As the number of prize winners has increased, a kinship has also arisen, where the previous winners have taken a real interest in being available and supporting the subsequent winners. I would just like to share with all of you, and particularly this year's winners, how very appreciative I am of all of you for the joy that this brings to me. Thank you.
Great. Thank you, Dr. Michelson. And now we have a few words about the prize from Dr. Wayne Koff, President and CEO of the Human Immunome Project.
Thank you. It's wonderful to be here. And I just want to echo the words of Gary. I think as we began this in 18, so it's been about a half a dozen years or so now, we were really interested in identifying the best ideas of the young investigators across immunology, vaccinology, AI, and machine learning. And in terms of this year's winners, this year was the most intensive of the competition of all the years we've had. We had hundreds of applications, and we had to triage those to a manageable number, about a dozen, and bring in another advisory group who looked at those. And we have the winners here, which you'll hear from in a minute. But I think an important issue is that if you're in your early stage of the career in science, in particular in the United States, the average age of the investigator-initiated grant is well over the age of 40. And yet, if we look back at the work of the winners of the Lasker Awards, of the Nobels, etc., in some of the best of the work is really done well under the age of 40. And so we really have an opportunity here to catalyze the work of extremely talented scientists across the whole range of immunological sciences. So I'll just stop there, and again I want to just go ahead and thank the Michelsons and congratulate all the winners on their incredible accomplishments this year. Thank you.
Great, thank you, Dr. Koff. Before we introduce this year's awardees in person, we'd like to share some remarks from a previous awardee, Dr. Danica Hill of Monash University, regarding the impact of the Next Generation Grant on her work and career.
Securing research funding is so important early on in your career. There's no universal pathway to transition from postdoc to running your own lab. And many institutions, they want to know that you and your ideas are fundable before they'll take that risk on you. But for many funding schemes, you need to be independent in order to be eligible to apply. And otherwise, in the very highly competitive funding landscapes that we face all around the world, often researchers just starting out on their own are viewed less favourably than very well-established teams. And so it can be really hard to attract that funding early on, and this can lead to many researchers feeling stuck, unable to make that next stage in their career. And that's why what the Michelson Prize does is so important. It provides that funding at that critical time that enables you to kind of make the next leap in your career, while also giving the prestige that winning an international competitive research prize brings as well. Winning the Michelson Prize in 2020 was so important for my career. It's improved my reputation internationally. It's led to invitations to speak at conferences, to author reviews. But really, it's gone a long way to proving that I can attract funding for my own independent research program, and was instrumental in my promotion to become group leader. So many, many congratulations to this year's 4 awardees. I'm sure this will accelerate your research just as it's done mine, and I encourage anyone listening to apply in the future. Thank you.
Fantastic. And now it's my pleasure to introduce this year's awardees who are working to push the limits of vaccine technology to advance towards intranasal delivery, single dose, and universal vaccines. They're developing new ways to examine microbes and immune responses for better vaccine development. After I introduce each awardee, we will show a 10 to 15-minute presentation about their work. And once all of our awardees have presented, we will have a collective group Q&A. You may submit your questions at any time into the Q&A box and vote on others' questions that you would also like to hear answered. Please note if your question is directed to a specific speaker in the text of your question. And now I'd like to welcome our first awardee, Dr. Noam Auslander, Assistant Professor in the Molecular and Cellular Oncogenesis Program at the Wistar Institute. Noam's research focuses on identifying microbes like viruses and bacteria found within different types of cancer and their impact on anti-cancer immune responses and patient outcomes. With the Next Generation grant, she aims to expand her work to examine microbes in immune diseases and their impacts on immunopathology and patient response to immunotherapies. Welcome, Noam.
Thank you so much, and thank you for the Michelson Foundation for this great opportunity.
Great. Noam's proposal is titled Artificial Intelligence-Based Identification of Microbes Associated with Immune Responses in Cancer and Immune Diseases.
My name is Noam Auslander. I'm an assistant professor at the Wistar Institute, and I received the 2022 Michelson Prizes for Artificial Intelligence-Based Identification of Microbes Associated Immune Responses in Cancer and Immune Diseases. I chose to become a scientist because I realized that I have high levels of energy and I'm interested in learning something new every day. So being a scientist, I can use those high levels of energy. There is always something to do in science. And I can also learn something new every day and even pursue new directions if I like to. So our research right now focuses on development of artificial intelligent methods to find new microbes in cancer and in immune diseases. We are finding those microbes that have not been discovered before in diseased tissues, and we are using them for better diagnosis of diseases and for better treatment strategies. The potential impact of finding new microbes on people's health is huge. If we find new microbes in cancer and in immune diseases, we can develop better early diagnosis strategies by knowing the microbes before the cancer develops, for example. If we find microbes that are associated with the disease development, we can improve prognostication and treatment decision-making. And if we find microbes that are associated with enhanced immune responses, we can use it as new treatment strategies as well. The Michelson Prizes are a huge help with my future research and career because they allow me to pursue the research directions that I'm most passionate about, which are considered high risk. It is not trivial that there are new microbes in cancer or in immune responses, and it is also not trivial that computational methods can help discover those. So it is not easy to get those projects funded by standard funding mechanisms, and the Michelson Prizes allow me to pursue those more high-risk directions and therefore potentially make very impactful discoveries based on those. Hello, my name is Noam Auslander. I'm an assistant professor at the Wistar Institute, and I will talk about some of our previous and ongoing work for this project, which is artificial intelligence-based identification of microbes associated with immune responses in cancer and in immune diseases, which is some of this work is what we received the Michelson Award for. And so I will describe the basis for this project, what we have been doing so far, and what we plan to do in the future with this award. So the general motivation for this is using artificial intelligence methods to find, to quantify microbes that are in diseased tissues, especially in cancer, and in next step we will also look into immune diseases. And the reason that we need artificial intelligence for that is that right now we are receiving sequencing data in this type of format that looks like that. We have a mixture of different organisms, very short reads from different organisms. For example, if we are looking into tumor sequencing or tumor RNA sequencing, we have mostly human reads probably, but then we also have some microbial reads, which can be bacterial or viral or other microbes, and some unknown reads that we cannot even characterize. And the idea, if we wanted to look into viruses or into bacteria, it is not very simple. It is not as simple as looking into the human genome, which is simple, which is, there is one human genome that we can map to. There are many different bacteria, there are many different viruses, and those organisms are highly mutating. And it is not a simple task to look at this mixture of different organisms and see what types of microbes are in our diseased tissues. So the idea is, if we have artificial intelligence-based method that can help us look into that complex mixture and distinguish the microbial reads and assist the process of quantification and characterization of which microbes are found in diseased tissues, we can do a much better job at that. And then we can eventually consider the microbes that are found in the diseased tissues when we are looking into disease immune responses. So the first project and the preliminary data that we have for these results, were specifically looking into viruses. We developed a method that is called ViroNetRep, which is identification of viruses and including new viruses that are expressed in tumors based on this rationale that I was just describing. And the idea is, we have very short reads from RNA sequencing. We can apply this artificial intelligence-based method, a neural network, a simple convolutional neural network, extract the viral reads, what are the reads that come from viruses, the viruses that are expressed in the tissue, or in this case, in cancer tissues. And then once we know those, we can get them into longer sequences and find known and also new viruses. So this work was led by Abdu, who is a postdoc in my lab, who developed this really great framework that can identify viruses and viral reads even with very short sequences. So Abdu first applied it to a data set to validate that he can do that. It was not trivial that with very short reads, as we often get for RNA sequencing, which can be even 48 base pairs, he was able to find, to predict what is a virus and what is not a virus with pretty good accuracy, and for different types of viruses. And the idea is that if we have this machine that can tell us what comes from viruses and what is not coming from viruses, we can potentially find even new viruses that we would not otherwise be able to find. So Abdu took his approach and applied it to data sets that are coming from tumors, to RNA sequencing from different types of tumors, and try to find what are the new viruses that we find with this approach. If we can extract the viral reads and get the longer sequences, do we find any new viruses? And in this case, we found many new candidate new viruses that were not reported in cancer before. And you can see they can come from bacteria, from different insects. Those are viruses that are usually associated with bacteria, or different insects, or plant viruses, and even mouse or bat viruses. For example, we found one candidate virus, which is an insect virus that was found in about 25% of uterine endometrial cancer samples. And this virus was surprisingly also associated with much better prognosis. So the presence of this virus in endometrial uterine cancer was linked with a much better prognosis of the patients. And when we looked at why are this new virus that was not reported before, why when we observe it, we see much better prognosis, we assumed it is probably linked to the immune responses. And when we looked, for example, at the T regulatory cells and at the CD8 T cells, immune T cells, we saw enhanced levels of those types of T cells in patients that were positive for the virus, which are confirming our hypothesis that probably this virus is associated with better immune responses, which is therefore linking this virus with better outcomes and better survival of the patients. So this is just a proof of concept that using artificial intelligence, we can find new viruses that were not previously associated with cancer. Those viruses can be linked to better immune responses, and therefore they can be used as biomarkers, as cancer biomarkers that can help us to better diagnose and better treat the patients. Another example of a new virus that we found, if we're looking at this, the second largest number is a virus that we found, a bacterial virus that we found in 33 samples of ovarian cancer that again was not reported before. And these ovarian cancers were expressing very different genes from this virus. And for this one, we also had an experimental validation. And we worked with Gang Zhang lab that was then at Wistar. And we try to validate whether we can see this virus in additional samples. And we looked at cell lines, and we were able to see that this virus is indeed expressed in ovarian cancer cell lines whenever we saw it with our approach. So that's a validation that some of the new viruses that we find, we can actually experimentally validate as well, which was a very strong result for us. So now the idea is that the work that is based on that and what we propose to do for the Michelson Foundation is to expand this work, the ViroNate trap that was originally designed to find only viruses, and to allow identification of bacteria as well, not just viruses, but also finding bacteria that are expressed in cancer and may be associated with immune responses and outcomes. And Daniel in my lab has been working on that, and he developed a model, an artificial intelligence model that can not only find viruses, but also find the bacteria that are expressed in disease tissues. And the idea is right now, if we have a mixture of all the different reads, we can extract the microbial reads, and then we can find longer microbial sequences as we did before for viruses. And then we can even see not just what are the types of bacteria that we see in tumors, but also what are the proteins, what are the bacterial proteins that are affecting immune responses or survival or prognosis in cancer and in immune diseases. So what are the goals of this project? We want to expand our artificial intelligence approach to allow identification and characterization of the microbes that are associated with cancer and also with immune diseases. We want to find, using these approaches, the microbes that are associated with outcomes and with enhancing immune responses. And another direction that we are also interested in looking into is to find whether we see specific types of microbes or microbial proteins that are associated with immunotherapy outcomes, because this is a factor that has not been considered so far. So overall, these types of approaches allow us to better characterize and quantify microbes in diseases and consider the presence of microbes as an additional factor that is underlying immune responses and leading to different prognosis and different development of diseases. So I would like to thank everybody in my lab, and especially Abdu, who led this project, and in Wistar, in the bioinformatics facility, Rugang's lab, who validated some of this for us, Alejandro at the NCI, and Quaid at MSK, who were great collaborators for this project. Everybody in my lab again, and of course, the Wistar Institute and the Michelson for supporting this research and making that this is really crucial for us and allowing us to pursue those more challenging directions. Thank you.
Thank you, Noam. Very interesting work. Our next awardee is Romain Guyon, a PhD student at the University of Oxford. Romain is developing an innovative extended-release vaccine delivery and manufacturing system to eliminate the need for vaccine boosters. The single-dose formulation provides the initial vaccine dose, along with doses that are harbored within the body until they're released from vaccine particles weeks to months later. Thanks for joining us, Romain.
Thanks very much for the opportunity.
His proposal is titled Demonstration of New Single-Dose Vaccine Technology Towards a First-in-Man Clinical Application.
My name is Romain Guyon. I'm a PhD student in the Jenna Institute in Oxford University, and I received the 2022 Michelson Prizes for demonstration of a new single-dose vaccine technology towards a first-in-man clinical trial. I was drawn to the field of vaccine development because it is such an amazing field that involves multiple scientific disciplines and can have a major impact on global health, both at a patient level but also at the scale of a population. My research focuses on the development of single-dose vaccines, and for that, I've developed a microfluidic-based technology to encapsulate vaccine booster dose inside microparticles that will deliver this booster dose after a certain time inside the body. This approach removes the need for multiple injections, which facilitates the vaccination process for the patient. The impact of my research is to improve vaccination coverage, in particular in countries where the access of medical facilities is limited. The aim is to provide a single-dose vaccine that would ensure complete immunization to protect people's lives. The Michelson Prize is a wonderful opportunity for me to establish myself as an independent researcher. It will give more visibility to the single-dose vaccine project and open more doors for collaboration. On the project, it will allow me to assess the compatibility of the technology with different vaccines and investigate new manufacturing strategies, taking it closer to clinic. I'm a finishing PhD student, both in the Jenna Institute and in the Institute of Biomedical Engineering in Oxford University. And today I'm presenting you my research focused on the development of a single-dose vaccine technology using microfluidics. I was awarded the 2022 Michelson Prize on the basis of that project. First, why single-dose vaccines? The answer is simple: to improve vaccination compliance. Usually, vaccines are given in a prime boost regimen, requiring multiple doses to achieve high immune responses and protection. However, this need for multiple injections at different times is a burden on the patient, particularly in low and middle-income countries, where access to healthcare is limited. To tackle this problem, we have worked here on the development of a delayed delivery system in the form of particles that could be injected alongside the prime dose in a single jab, remain at the site of injection for the required time, slowly degrading until the particles release their booster dose content. The particles we have created have a core shell structure, where the vaccine is contained inside an aqueous liquid core with excipients and adjuvants, and this core is surrounded by a solid shell of PLGA, a biocompatible and biodegradable polymer. We had multiple aims creating this system. First, the particles had to be small enough to pass through a needle and be injected, while being large enough to remain at the site of injection without being uptaken by any cells, thus having a diameter between 50 and 100 microns. They also needed to be uniform, with a very low coefficient of variation to make sure that the dosing and releasing is the same between particles. Finally, the kinetics of release from particles needed to mimic the booster vaccination, with delays that could be tuned to adapt the system to different vaccines. For that purpose, we have developed a microfluidic-based technology to manufacture this vaccine delivery system. Microfluidics is the manipulation of fluid flows inside tiny channels, where the small dimensions ensure laminar flow condition, which is essential for the emulsification process we are using to make the particles. For that, we have made these custom silicon chips you can see here, with a specific patented design. Let's take a look closer at the design. Here, you can see a microscopic view of the manufacturing process of the particles inside our microfluidic chip. The channels form two different intersections, one first intersection here and a second one there. At the first intersection, the aqueous flow containing the vaccine and excipients and adjuvants is injected and meets an immiscible fluid of solvent and PLGA polymer. This results in the formation of core droplets of vaccine solution. By the same emulsification process, the second intersection produces the outer shell of solvent and polymer, surrounding the core, forming what we call double emulsions. This is a video of the process running, slowed down more than 200 times. With this production process, we can collect the double emulsions and extract the solvent to harden the shell, forming our aimed core shell particles, which were very uniform. In this microscopic image, the core was labeled with a fluorescent payload for visualization. Using scanning electron microscopy, we can take a look at the mechanism of release. You can see right now a particle freshly produced with a very smooth aspect. Looking at its section, there is no sign of degradation, and the core is intact and fully formed. After 30 days of in vitro degradation at 37 degrees, you can see that the pores were formed inside the shell as a result of the degradation of the polymer. In the section, we can see a network of pores that was developed and interconnected. And the release occurred when the pore network formed a path between the inside core and the outside, triggering the release from this particle. Using an in vitro setup, we measured the cumulative release over time of the fluorescent payload from batches of particles, displayed here. We could first observe a lag period, where the particles kept their content and remained fully loaded. With the degradation of their shell, a sharp release was seen after 16 days for this particular formulation, where most of the particles released their fluorescent payload. With this specific lag before release kinetics, the particle system mimicked the booster vaccination, which was the initial aim. By changing the formulation of the polymer we used as the shell material of the particles, we could increase the delay before release. First, by increasing the molecular weight, the lag was pushed by a few days to a week. But also, by changing the lactic acid content, we could increase the delay by weeks and months, making this technology very suitable for different vaccination schedules. We did multiple in vivo experiments in a mice model as a proof of concept of the technology, and I'm going to present one of them. For this experiment, we used the malaria vaccine developed in the Jenna Institute in Oxford, who is currently in phase 3 clinical trials. We had 3 groups of mice in this experiment. The standard group received the classic prime boost vaccination regimen, with the first dose given as the prime and the booster dose injected 3 weeks later. The control group received the 2 doses of vaccine in the same prime injection. Finally, our group of interest received in one single injection both the prime vaccine dose in a soluble form and the booster dose encapsulated inside particles with a certain shell formulation. Looking at the antibody-specific response to the vaccination, both prime and prime boost groups behaved as expected. The antibody response was similar for the first 3 weeks. Then,
the antibody titers in the prime boost group increased by 10-fold following the booster vaccination effect. Our prime and particle group also had similar antibody titers for the first 2 weeks, before increasing significantly compared to the prime group due to the release of the particles, almost achieving the antibody levels of the prime boost. We also challenged these groups of mice with malaria to measure the protection given by each regimen. In the prime boost group, we saw 88% of protection, compared to the only 13% for the prime-only group. The prime and particle group protection was 45%, not reaching the level of the prime boost efficacy, which could be explained by a suboptimal timing of release or the lower dose of adjuvant encapsulated in the particles. However, the prime and particle increased by more than 3-fold the protection of a single injection vaccine when compared to the prime. This experiment showed the ability of the particles to improve single injection formulations of vaccines and demonstrated the potential, with some adjustments of the formulation, to match the immunogenicity and protection given by prime boost regimen. Following these results, I was awarded the 2022 Michelson Prize for the demonstration of a new single-dose vaccine technology towards a first-in-man clinical application. With the great support of the prize, I will now focus my research on 2 specific aims. First, demonstrating the use of the microfluidic-based core shell particle technology for delivery of a licensed, commercially available and well-established rabies vaccine, with the aim of facilitating a future clinical trial. The second aim would be to develop the current prototype platform to demonstrate the feasibility of scaled-up manufacturing. Why rabies? Rabies is still nowadays a deadly infection that kills more than 60,000 people per year across the world, despite an existing effective vaccine working both pre- and post-exposure. And one of the main reasons for this death toll is the difficulty of access and compliance to the rabies vaccine regimen. For the post-exposure prophylaxis schedule, 4 vaccines injections are required to achieve high neutralizing antibody titers, given at day 0, 3, 7, and 14. Our aim here would be to replace this regimen by a cost-effective single injection containing both the prime vaccine and vaccine-loaded particles. To develop this approach, the compatibility of the rabies vaccine will be first tested to ensure the stability inside the particles and the maintenance of immunogenicity. Then, in vivo experiments in mice will be conducted to optimize the release and reach the threshold of protective neutralizing antibody titers. The second aim that the Michelson Prize is allowing me to pursue is the increasing of the manufacturing throughput. The microfluidic platform developed is currently able to encapsulate 4 doses of vaccine per hour. We're aiming to multiply this production rate by a minimum of 10 within our lab setting, reaching more than 40 doses per hour to demonstrate the scale-up potential of the system to achieve batch production that could be used in human trials. For this objective, different fluid formulation will be changed to accelerate the emulsification process inside the particles. The vaccine will be concentrated before encapsulation, and parallelization designs of the microfluidic chip will be tested. You can see here one of the parallelized designs that will run 4 different emulsification processes at a time. Finally, new sets of fluidics and monitoring equipment will also be purchased to partially automate the platform. I would like to thank everyone that contributed on this project, from the Jenna Institute and the Institute of Biomedical Engineering, and in particular my PhD supervisors, Anita, Adrian, and Eleanor. I would also like to thank both the Michelson Foundation and the Human Immunome Project teams for the amazing opportunity they gave me to push this project forward after my PhD. Thank you.
Thank you, Romain. Very exciting work. Next, we have Dr. Brittany Hartwell, Assistant Professor in the Department of Biomedical Engineering at the University of Minnesota. Brittany's work focuses on how to get vaccines to generate mucosal immune responses that protect against viral infection at sites of entry, like the nasal passage and the lungs. By investigating techniques for intranasal delivery, she aims not only to provide easier administration of vaccines without injections, but also to induce better mucosal immune responses at the most vulnerable sites. Thank you for joining us, Brittany.
Hi, thanks so much for having me, and thanks for the opportunity.
Brittany's proposal is titled Engineering Albumin Hitchhiking Intranasal Vaccines with Enhanced Transmucosal Uptake to Promote Protective Immunity.
My name is Brittany Hartwell. I'm an assistant professor of biomedical engineering at the University of Minnesota, and I was awarded the 2022 Michelson Prizes for my work on engineering albumin hitchhiking intranasal vaccines with enhanced transmucosal uptake to promote immunity. I was drawn to the fields of biomolecular engineering and immunology due to my combined interest in medicine and engineering problem solving. It was as a graduate student that I became fascinated with immunology, the sophistication and interconnectivity of our immune systems, and especially the possibilities for using engineering and drug delivery to intervene and direct the immune response. Overall, my lab's research in immunoengineering combines perspectives from biomolecular engineering, drug delivery, and immunology to develop safer and more effective immunotherapies and vaccines. To combat long-standing epidemics such as HIV and persisting threats such as SARS-CoV-2 and its continually emerging viral variants, immunization strategies are needed that elicit immune protection at mucosal portals of entry to halt transmission. However, traditional injected vaccines typically elicit poor mucosal immunity, and only a small number of mucosal vaccines have reached licensure, most of which are based on live attenuated pathogens. And this is largely due to poor uptake and drug delivery across mucosal barriers. Thus, our work on developing albumin hitchhiking intranasal vaccines that can more efficiently deliver vaccine components across mucosal barriers has the potential to address an urgent unmet need for effective mucosal vaccines against a broad range of infectious diseases. The Michelson Prize will help with my future research and career by providing critical funding to help launch my independent lab's research on intranasal vaccines that use this strategy of albumin hitchhiking for enhanced uptake across mucosal barriers. Importantly, the recognition of this award will bring exposure to the significance and relevance of mucosal vaccine research and to the work my lab is doing in this area.
Hi, my name is Brittany Hartwell. I'm an assistant professor of biomedical engineering at the University of Minnesota, and I'm thrilled to have this opportunity to share my work for the 2022 Michelson Prize on engineering albumin hitchhiking intranasal vaccines. To combat long-standing epidemics such as HIV and persisting threats such as SARS-CoV-2, we need immunization strategies that can elicit protection at mucosal portals of entry. Transmission of many pathogens, such as HIV, SARS-CoV-2, influenza, and CMV, occurs primarily by mucosal routes, requiring both systemic and mucosal immune responses for effective protection as a front line of defense. Yet, most traditional parenteral vaccines that are injected are actually not great at activating immunity in mucosal sites of transmission. By contrast, mucosal vaccines that are administered directly at barrier tissues can promote mucosal immunity, and can even promote mucosal immunity at multiple different sites through a mechanism known as common mucosal immunity. Activation in the nose, for example, can promote immunity in distal sites like the genitourinary mucosa, as well as local sites like the respiratory tract, by triggering homing of immune cells. This illustrates how we could use this shared route of intranasal administration to protect against pathogens with very different routes of transmission, like HIV and SARS-CoV-2. Yet, although well motivated by the biology of mucosal immunity, delivery of vaccine components across mucosal barriers has been a major limitation for mucosal vaccine development, and as a result, only a few are licensed for use in the clinic. Uptake into the underlying mucosal immune compartment is impeded by multiple factors, such as mucociliary clearance, antigen degradation by enzymes and acidic conditions, and a lack of diffusive uptake across the epithelial monolayer. But while mucosal barriers are very good at keeping most vaccine components out, we know that albumin, a naturally occurring serum protein found in mucus as well as the blood, is actually very good at getting in. And it does this by binding to the neonatal Fc receptor, or FcRn, which is expressed on mucosal epithelial cells. And this transcytoses albumin across mucosal epithelium into the underlying submucosa. We also know that albumin acts in vivo as a fatty acid transporter, bearing multiple lipid binding sites. So we thought, what if we can create a vaccine that takes advantage of these lipid binding properties and uses albumin as a chaperone to effectively hitchhike across mucosal barriers to get into the NALT, or the nasal-associated lymphoid tissue. This is where antigen-specific immune responses are orchestrated. By concentrating vaccine in the NALT, it can better activate key immune cells like B cells and T cells, which then mobilize a frontline defense of resident immune cells in the mucosal tissue, along with mucosal antibodies that can neutralize or otherwise stop virus before infection takes place. The overarching goal of my Michelson Prize work is to explore this strategy as a way to create better infectious disease vaccines. And it's based on previous work that I carried out as a postdoc in Daryl Irvin's lab at MIT. So I'd like to quickly acknowledge this group, my lab mates, my previous advisor, Daryl, and our collaborators. I first set out engineering protein amphiphile vaccines that could protect against HIV with a goal of activating mucosal humoral immunity in the genitourinary tract where HIV is transmitted. I was using a model B-cell protein antigen derived from the HIV glycoprotein envelope called GP120 or EOD. I conjugated this B-cell protein antigen onto an amphiphile platform consisting of a lipophilic diacyl tail, which was conjugated to a hydrophilic polyethylene glycol linker. And this amphiphile tail endowed the molecule with the ability to bind albumin, exploiting albumin's role as a fatty acid transporter. I hypothesized that this would enhance transmucosal uptake by hitchhiking on albumin. So my first question was, does amphiphile vaccine actually promote uptake and retention in the nasal passage? We evaluated this by fluorescently labeling amphiphile vaccine, or AMPH-EOD, and soluble EOD, which is our control soluble protein, and intranasally immunizing mice. We then looked at the vaccine signal using IVIS, or in vivo imaging, in the nasal cavity of the mouse, which is shown here. You can see strikingly that the EOD soluble protein was cleared within 24 to 48 hours. In contrast, the amphiphile vaccine persisted out to 11 days. There was actually no significant reduction in signal over 3 days, and by quantifying this signal, we saw there was significantly greater total vaccine accumulation in the nasal cavity as a result of this simple amphiphile modification. Notably, when we repeated this study in FcRn knockout mice that lacked expression of the neonatal Fc receptor, we again saw significantly reduced uptake of our vaccine, down to levels that we saw with the soluble protein, supporting this mechanism of FcRn-dependent uptake. We then wanted to take a closer look at the actual uptake across the mucosa into the epithelium and underlying submucosa. So we took histological cross-sections of the mouse nose and evaluated vaccine uptake by fluorescence microscopy. So you can see the mouse nose cross-section here, with the vaccine shown in red. This middle row is the amphiphile vaccine in wild-type mice. You can see at 6 hours, there's significant vaccine deposition on the epithelium labeled with E, and then quite a bit of uptake into the underlying lamina propria. You can see these little vesicles as the vaccine is being taken up. And this is even more clear when you go out to 24 hours, take a closer cross-section, and you can see the vaccine not only on the epithelium, but here being transcytosed into the underlying lamina propria. In contrast, the soluble protein, EOD, was mostly trapped in the airway, in the mucus shown here in M, and by 24 hours it was completely absent. Interestingly, the amphiphile vaccine in FcRn knockout mice still deposited on the epithelium, but taking a closer look at 24 hours, although we see it on the epithelium, it's notably absent in the lamina propria because it lacks this FcRn uptake. So we were seeing enhanced FcRn uptake. Our next question was whether this uptake was actually leading to concentration of the vaccine in the NALT, or the nasal-associated lymphoid tissue. This is a secondary lymphoid tissue located here at the base of the nasal passage, also located here in 2 strips on the underside of the mouse nose, underside of the palate. This is where antigen-specific immune responses are orchestrated. We saw enhanced uptake in key antigen-presenting cell populations like macrophages, B cells, and dendritic cells in the NALT. And this led to enhanced germinal center responses in the NALT in antigen-specific germinal center B cells, total germinal center B cells, and TFH cells, T follicular helper cells, in an FcRn-dependent manner. This is important because the germinal center is a training ground for a high-affinity antibody response, so activating these cell populations is a critical precursor to a downstream humoral antibody response. which we then evaluated using this longitudinal study, immunizing mice intranasally with AMP-EOD versus EOD. So here we follow the systemic immune response over 35 weeks, looking at serum IgG versus serum IgA. And you can see that with our amphiphile vaccine, we saw significantly enhanced responses, both in the serum, but importantly, also in the mucosa at clinically relevant sites for HIV transmission. So in the vaginal and rectal mucosa, we saw significantly enhanced IgG and IgA over 35 weeks. Over 1 year post-immunization, we still saw the presence of antigen-specific IgA-secreting plasma cells resident in the vaginal mucosa, which indicated that we had actually promoted bona fide long-lived resident immune cells in mucosal tissue with our amphiphile vaccine, which was really encouraging. We next sought to apply this vaccine strategy to COVID, developing a vaccine against SARS-CoV-2 consisting of this amphiphile platform conjugated to the receptor binding domain antigen, which is the portion of the spike protein of SARS-CoV-2 that binds to the ACE2 receptor. And in this case, our goal was to activate mucosal immunity, not only activate systemic immunity in the blood, but also mucosal immunity in clinically relevant sites for SARS-CoV-2, meaning the upper and lower respiratory mucosa. We evaluated this looking at IgG and IgA antibodies that bound to RBD. You can see here striking enhancement of antibody responses in the serum, and also, importantly, in these clinically relevant sites in the saliva, the nose, and the lungs. Even more exciting was our neutralizing antibody results, where we saw over 1,500-fold greater neutralizing antibody responses in the serum from our amphiphile vaccine, and enhancement in the nose and the bronchoalveolar lavage fluid, or the lungs. I think what's really exciting here is this over 50-fold enhancement of neutralizing antibodies in the nose from the amphiphile vaccine, because to date, this has been a really challenging site to activate neutralizing antibody responses of other COVID vaccines in development. So I think this speaks to the translational potential of this vaccine strategy for a disease like COVID. These results suggest that employing AMP vaccines to deliver antigen across the mucosal epithelium is a promising strategy to promote mucosal immunity against HIV, SARS-CoV-2, and other infectious diseases. Going forward, the overarching goal of my Michelson Prize work is to continue developing and investigating this molecular platform and mechanism of FcRn-mediated uptake to create better infectious disease vaccines. We'll start by synthesizing a library of candidate amphiphile vaccines with different types and sizes of immune cargo. Then evaluate transmucosal uptake using imaging in parallel with mucosal immune responses. And finally, correlate these properties and behaviors with functional immune protection. To date, this platform has been investigated with HIV and SARS-CoV-2 antigens that are both protein monomers of about 22 kilodalton. Investigating this platform now with antigens of varying molecular weight, ranging from peptides of about 1 kilodalton to protein trimers of a few hundred kilodalton, and defining the compatible antigen molecular weight limits, will expand the translational potential of this platform to a range of other diseases. Conjugating adjuvants as well into the amphiphile platform to facilitate spatial and temporal co-delivery of antigen and adjuvant should also improve the safety profile and efficacy of this vaccine strategy. We'll then evaluate transmucosal uptake using the imaging methods I covered earlier, with a goal of defining this relationship of intranasal FcRn-mediated mucosal uptake as a function of cargo molecular weight. In parallel, we'll assay multiple aspects of the mucosal immune response, including looking at germinal center responses in the NALT, resident antigen-specific B-cell, plasma cell, and T-cell responses in clinically relevant mucosal tissues, and antibody responses in the mucosa. Microbial environment also plays a significant role in the mucosal immune response, yet is often overlooked in preclinical development of mucosal vaccines, as studies are carried out by default in specific pathogen-free or SPF mice. Thus, we'll evaluate these responses in both clean SPF mice and dirty mice, using the University of Minnesota's unique dirty mouse colony core facility. Lastly, we'll evaluate functional immune protection using a mouse model of SARS-CoV-2. I hypothesize that enhanced transmucosal uptake of AMP vaccines will lead to greater functional protection against viral challenge by establishing resident immunity in the upper respiratory tract. And that promotion of strong germinal center responses in the null will also generate higher affinity and greater breadth of antibody responses. To test this hypothesis, we'll investigate the ability of candidate COVID vaccines to induce functional protection against viral challenge with multiple variants in the K18 mouse model. To conclude, I propose that conjugation of antigens with albumin-binding lipid moieties presents a simple and robust approach to overcome a major obstacle to the development of mucosal vaccines, that is, uptake across the mucosa. This immunization strategy has the potential to significantly improve vaccination efforts against a broad range of diseases. With that, I'd like to thank or acknowledge my newly founded lab here at the University of Minnesota, the Hartwell Immunoengineering Lab, and my lab members. I'd like to especially thank the Michelson Medical Research Foundation and the Human Immunome Project for funding this work and for giving me the opportunity to present today. Thank you so much.
Thank you, Brittany. Very impressive. And our final awardee today is Dr. Jenna Guthmiller, Assistant Professor in the Department of Immunology and Microbiology at the University of Colorado Anschutz School of Medicine. Jenna studies how to generate antibodies that protect against multiple different types of influenza strains rather than individual seasonal strains to enable a universal flu vaccine. She has developed a human tonsil organoid system to study immune responses to vaccine candidates in the laboratory. She aims to utilize this system to determine how to generate durable and broadly protective immunity against influenza infection at mucosal sites. Thanks for joining us today, Jenna.
Thanks for having me.
Jenna's proposal is titled Mapping Pre-Existing Mucosal B-Cell Specificities Engaged by Potential Universal Influenza Vaccines.
My name is Jenna Guthmiller, and I am an assistant professor within the Department of Immunology and Microbiology at the University of Colorado Anschutz Medical Campus. I received the 2022 Michelson Prize with my proposal entitled Mapping Pre-Existing Mucosal B-Cell Specificities Engaged by Potential Universal Influenza Vaccines. My passion for immunology stems from my childhood, where one of the earliest memories I have was vaccinating animals on my family's dairy farm. The idea that we could give a dead or weakened version of a pathogen that could provide protection against something that would otherwise make the animals sick was fascinating to me and has left a lifelong impact on my desire to understand how to limit infectious disease burdens. My research group is investigating how broadly protective immunity develops against influenza viruses and how vaccines can preferentially and robustly induce these protective responses. The current project is focused on dissecting how immunity within the respiratory tract differs from that within the blood, and how potential universal influenza vaccines can engage broadly protective immunity within the respiratory tract. The impact of our research is that we will provide a better understanding of the antibody specificities within the respiratory tract and how to optimize vaccines to induce these protective immune responses. This study has direct implications for human health, as it'll inform how to design mucosal vaccines that can provide protection at the site of influenza virus infection. The Michelson Prize allows me to expand our research into the field of mucosal immunology, which will undoubtedly lead to new research directions within my laboratory. As is, our current understanding of mucosal antibody responses against respiratory pathogens is limited. This prize allows us to develop new tools in collaboration with clinicians to study antibodies against influenza viruses at mucosal sites, which will help move our science from the bench to the bedside. All right, it is my pleasure to present on my research, which is mapping pre-existing mucosal B-cell specificities engaged by potential universal influenza vaccines. And so my name is Jenna Guthmiller. I'm an assistant professor within the Department of Immunology and Microbiology at the University of Colorado Anschutz Medical Campus. And so my lab studies humoral immunity against influenza viruses and why it's so difficult to generate broadly protective immune responses. And there's several factors that really inhibit our capacity to generate broadly protective humoral immunity or antibody responses. And one of these is that influenza viruses are just incredibly antigenically variable. And what I mean by that is there are multiple types that can infect us and cause significant morbidity and mortality, and those are influenza A and influenza B viruses, with influenza A made up of multiple subtypes and influenza B made up of multiple lineages. In addition to that, influenza viruses undergo this process known as antigenic drift and shift, with antigenic drift just being the process of influenza viruses mutating to evade our antibody responses, whereas antigenic shift is the reassortment of multiple influenza virus genomes to generate antigenically novel influenza viruses. And so all I'm highlighting down here is that there's a lot of complexity when it comes to the types of influenza viruses that can infect humans, potentially. In addition to that, when we do generate an antibody response against influenza viruses, we're faced with this issue of immunodominance, meaning that we do not target all parts of the virus equally, with us preferentially targeting variable epitopes, so those that like to mutate a lot versus those that are well conserved. And so on the right-hand side, I am highlighting the influenza virus protein hemagglutinin, or HA. And what I really want you to appreciate here is that that head domain is much more variable than that of the stalk domain. And for whatever reasons, we preferentially generate antibody responses against those variable epitopes of the head domain and not so much those conserved parts of the stalk domain. Furthermore, we preferentially generate responses against non-protective epitopes of viruses, rather than protective epitopes. And cumulatively, we tend to generate very few broadly protective antibody responses. And so together, these two factors can really contribute to the low seasonal vaccine effectiveness that we currently have with influenza vaccines. And so, as I mentioned, we do have strain-specific vaccines. So this is what we would receive every fall or in our respective countries. And these are really meant to target current circulating strains, whereas a more universal influenza virus would target more strains within a subtype or multiple subtypes at a time, or both groups of influenza A or both influenza A and influenza B viruses. And as antibodies against HA are a known correlate of protection, it indicates that broadly protective antibodies are really our answer to generating a more universal flu influenza vaccine. And so, as I mentioned before, again, that stalk domain across H1 viruses across group 1 or group 2 viruses is much more well conserved than that of the head domain, suggesting that the stalk is a good target of antibody responses to generate a more universal flu vaccine. But the questions that my lab is interested in addressing are: what are the specificity, function, and breadth of these broadly protective antibodies that can be induced, and how can vaccination preferentially induce these broadly protective antibodies? And so we've previously shown for H1N1 viruses that there are 5 broadly protective epitopes, 3 on the HA head, including the receptor binding site in the lateral patch, as well as 2 on the stalk domain, so the central stalk and anchor epitopes. And so how we identify antibodies against these particular epitopes is that we take B cells from humans after influenza virus infection or vaccination. And so what's happening is that as adults, we all have some level of pre-existing immunity against these viruses. And when we get infected or vaccinated, we can have this B cell population, or these memory B cells, turn into plasmablasts, which is a short-lived antibody secreting cell that pops up in the blood about a week after these exposures. On the other hand, we can also pull out memory B cells at later time points to understand how these memory B cells have changed following infection or vaccination. Again, really focusing on these 2 B cell populations. And from these populations, what we can do is generate monoclonal antibodies. So 1 B cell equals 1 monoclonal antibody. And so what this allows us to do is to interrogate epitope specificity, breadth, and function of B cells that are induced by these various exposures. And so the question, again, going back to, is how can vaccination recall memory B cells against these broadly protective epitopes? And so, again, I will keep showing you this until you can remember it, is that the stalk domain is pretty well conserved, whereas that of the head is not. And so the question that we had is that if we have low pre-existing immunity against these variable epitopes, which means we don't have very many memory B cells against those epitopes, can we preferentially recall memory B cells against these conserved broadly protective epitopes? And in order to do that, we had multiple cohorts of individuals with low or high pre-existing immunity against the 2009 H1N1 virus. So again, low pre-existing immunity in this case means it was their very first exposure to this virus, or high pre-existing immunity meaning that they had already been exposed before. And what we find is if it's their very first exposure to this virus, that they're much more likely to generate a stalk antibody response than those that have been repeatedly exposed to this pandemic H1N1 virus in 2009. Moreover, if we looked at these broadly conserved and protective epitopes of the HA head domain, so the RBS and the lateral patch, a similar story. If individuals have low pre-existing immunity against these epitopes, variable epitopes that is, that they're much more likely to recall memory B cells against these broadly protective epitopes of the head domain, as well as the stalk domain, as I'm showing over here. Okay, so with this knowledge, our colleagues at Mount Sinai designed a vaccine to kind of exploit this process of low pre-existing immunity against these variable epitopes. So as I mentioned before, we have oodles amounts of pre-existing immunity against H1N1 viruses. And the idea being that if we change that head domain to something that we have no pre-existing immunity to, so in this case, exotic HAs from avian influenza viruses such as H8 or H5, and you give sequential boosters of these chimeric HA antigens with the same stalk domain, can we preferentially induce antibody responses against the stalk? And lots of preliminary data has shown that this does work. So as a result, this moved into a phase 1 clinical trial a few years ago that we are fortunate to be a part of to help understand what are the specificities of the antibodies that can be induced by this chimeric HA vaccine approach. So just to note, if we look at serum antibody responses following this vaccine in humans, there were multiple formulations, but just to note that those that received an inactivated vaccine plus adjuvant intramuscularly, that they were able to induce a robust antibody response or serum antibody response, so in the blood, against the stalk domain. And so the question that we came into this was, does chimeric HA vaccination induce antibodies against those broadly neutralizing epitopes of the HA stalk that I had mentioned previously? And so in order to do this, I worked with a technician in my postdoctoral lab, Henry, in order to do these serum competition assays with antibodies against known epitopes of the stalk domain. So we can actually quantify what are the levels of antibodies against broadly neutralizing antibodies within the serum. And so this is just a lot of data to show what the vaccine trial looked like, but just to jump into the data, what we found was that individuals on both the prime and the boost, that they were seeing the inactivated vaccine with this oil and water adjuvant were much more likely to generate these broadly neutralizing antibodies against that anchor epitope, as well as that central stalk epitope. So this is shown for both the prime here, as well as the boost, indicating that the chimeric HA approach can induce broadly neutralizing antibodies within the serum, at least, against these broadly neutralizing epitopes. So what are the take-home messages so far? The first is that memory B cells to these broadly protective epitopes are actually quite abundant and that they can be recalled in the absence of competing head antibodies. So we've shown that with two different systems, the first being that the 2009, first exposure to that 2009 H1N1 virus, as well as the chimeric HA vaccine, albeit in the context of this adjuvant, because without the adjuvant, they don't generate much of a stalk antibody response relative to that with the adjuvant. But one thing I didn't really go into is that all of the vaccines that we study, for the most part, are given intramuscularly or IM. And as a result, these responses in the blood and the measurement of responses in the blood may not be predictive of what is actually happening in the respiratory tract. And as flu is a respiratory virus, it makes a lot of sense that we want to know what's not only going on within the respiratory tract, but what are the protective antibody responses that can be generated at the site of infection? And so the question we had, because it's incredibly difficult to study these things in humans, is how can we model how humans will respond to vaccination? And so what we have been doing is we've adapted an ex vivo tonsil organoid culture where we can get tonsils from individuals, or in this case, we're adapting this to lung draining lymph nodes. And what we do is we chop up all of these tissues, and when we're ready to study them, we can plate them into a transwell system, stimulate them with antigen, and start to look how B cells are responding to the antigen that we have given these cultures. And so just a note, what we've done so far is that if you give these tonsil organoids the seasonal influenza vaccine plus an adjuvant in this case, relative to just recombinant protein alone, that the vaccine is much better at inducing what we call antibody secreting cells, the CD27 high, CD38 population that I'm highlighting here. So again, we can use this system to understand how vaccines can induce antibody responses in the lymph nodes that are associated with the respiratory tract. Moreover, when we think about, these next generation or universal influenza vaccines, how do we preferentially generate this durable and broadly protective humoral immune responses at these mucosal sites? And unfortunately, I don't necessarily have data for this, but I just want to highlight the things that we are doing as a result of the funding from this Michelson Prize. And so the major questions that we have are, what are the B cell specificities within the lungs? So how are the specificities that we've developed within the lungs and these lung draining lymph nodes distinct or similar to that of what is currently circulating? And so what we have been doing is we've been collecting tissues from people of various ages and started to pull out those B cells to understand and make monoclonal antibodies in order to understand what parts of the virus that they are preferentially targeting. In addition to this, we can take those tissues and address the question of which B cell specificities are engaged by these potential universal influenza vaccines that are trying to generate responses against the stalk domain. And so using that organoid system, we can model the humoral immune response against these vaccine candidates to really address how robust humoral immunity can be generated within the respiratory tract. And so with that, I just want to thank the people of my lab that have started to work on this project, as well as our collaborators, Sarah Clark, Patrick Hume, and then Lucia at the University of Chicago, in their capacity to provide us with various tissues to start to address these questions, as well as all of the other collaborators that have made this data possible, our funding, and any questions that anyone may have for me. Thank you.
Okay, great. Thank you all for joining us today to share your inspiring work. Now I'd like to welcome all of our panelists back on screen. And we will do our audience Q&A. So for the audience, again, submit your questions into the Q&A tab, and you can vote on others' questions so we can gauge audience interest. We already have a few in there. I'll start by asking our first question to Romain. We have an audience question. Do you think the slow release methodology is amenable to multiple types of vaccines, such as subunit, adjuvant, replication defective virus, and mRNA vaccines?
It's a good question. So far, I've tested a lot of protein-based vaccines, viral vectors. So all this is relatively fine. One big key thing about the technology we're working here in Oxford, and it's intrinsic to the principle of having a single dose vaccine, is that your particle will stay inside the body at body temperature, so 37 degrees. So something like mRNA will probably not be stable because it's already not stable at room temperature. And we've seen that it was quite a big burden of logistics to keep it at the correct temperature during the pandemic. So it's probably compatible with most of the existing vaccines so far, but the mRNA technology.
Got it. Thanks. And for Noam, what do you expect to find with your studying immune diseases? Do you expect to find a role of microbes in any particular immune diseases above others?
So we have been starting to look into multiple sclerosis with collaborators, for example, because recently EBV has been demonstrated to be activated in almost 100% of those patients. So we are looking there, not just at EBV, but whether we find other viruses and maybe bacteria. And in general, I think that's a proof that probably there is a lot of viruses and bacteria in other diseases as well that are just not studied. So once we have enough resources for that, we will just look into every data that is out there and with the approach that we already have and those that we are developing and see whether we find any new associations.
Mm-hmm. And for multiple sclerosis, I know some of those are immune generated, some are more autoimmune generated. Do you feel autoimmune diseases will have a particular role of these microbes or not?
So we don't know. That was a pretty EBV and multiple sclerosis has been, the link has been known for a while, but demonstrated to that extent of the activation just very recently. We don't know. However, there are findings that connect viruses and many other diseases as well, diabetes, and there are really many other types of diseases that has been associated with viruses, bacteria as well. So I think I don't know whether it's a more autoimmune or just other types of immune diseases, but yeah.
Okay, great. And we have a question for Brittany. Could you conjugate antigens to albumin rather than relying upon the fatty acid interaction with albumin?
Yeah, that's a good question. You could do that. Actually, people have explored that as a strategy for direct albumin hitchhiking, covalently conjugating. However, I think there are advantages to instead conjugating to this amphiphile tail. One is that this amphiphile tail also inserts into the cell membrane. This is kind of a secondary property that helps its retention in lymphoid tissues. And also, if you remember from my talk, we saw even in the FcRn knockout mice that we had some retention of the amphiphile vaccine in the epithelium. And we proposed that that was one reason. So by having this amphiphile tail, the trafficking, once it gets to the submucosal layers and any other underlying lymphoid tissues, is independent of any subsequent albumin trafficking. It can dissociate and then better be retained in that tissue. And there are some disadvantages with having persistent trafficking with albumin when you're trying to design a vaccine, is that albumin is then going to be recycled and distributed systemically, and you don't want a vaccine to go systemic. You want it to stay in that lymphoid tissue to maximize the immunogenicity and the safety of the vaccine.
Mm-hmm. Great. And Jenna, can you see a way to apply your technique for other viruses like SARS-CoV-2, which also we're having issues with kind of mutating different strains and things, and even beyond that?
Yeah, absolutely. So I didn't mention it, but of course, with these technologies, we're also applying RSV, SARS-CoV-2, and human metapneumovirus into our analyses. Because we have these tissues, we really want to make sure that we're exploring all of the different specificities that we can find within these lung-draining lymph nodes. Because of course, and I think Brittany's talk really highlighted this, is that, mucosal vaccines are a great opportunity to really combat these diseases, not just flu, but many others.
Great. And for all of you, actually, how could you apply your techniques and strategies for broader, whether it's different types of viruses or even cancer vaccines? Can you kind of expand on how your methodologies might be applied to other vaccine development?
I could go first.
Great.
So my lab really focuses on isolating B cells and figuring out what they're specific to. And that can really be applied to any context where we are trying to pull out B cells in a disease specific context. And so, it could be cancer, it could be other pathogens, it could be a variety of different things. And a matter of just getting these kind of protocols and techniques working in various contexts has a lot of application to pretty much anything you can imagine.
Great. Romain?
I'll go next.
Okay, great.
Yeah, go.
Yes.
Okay. So my lab works on developing vaccine and immunotherapy platforms that, in essence, can be translated to multiple different diseases by just swapping out that immune cargo that we're trying to deliver. So in this case, it would be the antigen that's conjugated onto our albumin hitchhiking amphiphile tail. So I talked about this a little bit in my future directions of my talk, but we're looking at conjugating different sizes of antigens now to determine what these design parameters are of the vaccine. And once we figure out those molecular weight limits, that'll allow us to start conjugating other types of antigens as well. We know that antigenic epitopes for other diseases aren't always nice protein monomers like GP120 or RBD. So once we start looking into that, then we can also start looking at activation of mucosal immune responses in other tissue sites, and that'll make this pretty broadly translational.
Great. And Romain?
For me, it's a bit a similar response. Like it's quite, I can adapt the technology to broader vaccines also, because essentially what we're working here is a wrapper. It's an envelope for anything, and because the vaccine is contained into this liquid core, whatever you already have injected as a prime vaccine, you will put it inside this core and surround it with your polymer shell that will degrade. So anything that could benefit from a conversion from a prime boost regimen to a prime only could be going into this technology, as long as they are compatible with the temperature constraint, which is the main limitation so far. But that can be worked separately by trying to stabilize it with some stabilizer.
Great. And kind of building along those lines, would there be a way to combine all of your approaches to kind of make a one dose intranasal universal vaccine?
I'm not specialist about intranasal. I know that the particles I'm making are quite big, so I think it might be one of the limitations for other routes than injection. They're not too big, so they can be injected, but probably I guess the retain, because the point is to keep these particles for a certain time before they release their contents. So make sure that they are retained into the mucosal sites, and injection might be a limitation, but we can talk more about this.
Yeah, Romain, I think your technology is super cool. I don't know if it would be compatible with intranasal delivery, but the idea of that controlled release and sustained release of a boost dose is super neat, and I think really like broadly applicable. Jenna, I think Jenna and my research is very complementary. I'm excited to talk to you more about that, Jenna. But and then of course there's a lot of overlap between like bacteria, other viruses and other diseases, like with Noam's research.
Yeah, and I guess just to in addition, my lab is really focused on these immune responses as a result of vaccination. So of course the application of Romain and Brittany's work is very applicable. So I'm glad to talk to them further about anything.
Exciting, we'll get some collaborations going here. We have another question for Noam from the audience. I'd like to hear more about the AI programs that you're using, how you develop those, what the challenges are in developing them, and kind of strategies moving forward there.
So we use convolutional neural networks, nothing too fancy, because it worked best. We tried different things. The main challenge is the sequencing length, the reads length of the sequences. If we want to find either bacteria or viruses at the read level, and then we can look into that. The current Illumina sequencing can be anywhere between 50 to 150 base pairs. The shorter it is, the worse it we're doing. For example, for bacteria, we figured out that we cannot find bacteria and viruses with the 50 base pairs. We need at least 75, 76 to do that well. So I would say the technology is there. We can develop a pretty good model to find those, but we are not able to do that with very short reads. So the development in Illumina sequencing and getting longer reads is definitely helpful.
Great. And we have a question for Brittany and Romain. What is your response when the general public expresses hesitancy about vaccines lingering longer before they're cleared, for example, in young children or pregnant people or anyone else who feels particularly vulnerable? And I think, Brittany, you can probably weigh in on that as well.
I guess I'll go first. Yeah, that's a good question. And there's, of course, been a lot of this over the last few years with the COVID pandemic. My response typically is just trying to talk about the underlying immunology basis to vaccines and how they work, and they're training our immune systems, and how the process for getting vaccines approved actually is very thorough so that they're safe. In terms of increasing retention times and half-life of vaccines in vivo and whether that's a safety concern, again, I would emphasize that a lot of preclinical and clinical studies are done to ensure that these are safe before they would actually be put into people in a clinic. And so far in our studies, we haven't seen any adverse effects from this sort of nasal vaccine just staying here. The basis, the amplified platform is pretty inert. We don't see a lot of antibody responses against any part of that amplified platform. It's really just getting the antigen into the lymph node. And you need that to stay around for a while in order to maximize an immune response. And I think one of the advantages of this type of intranasal vaccine is that it is actually safer and can be used in more of the population than something like a live attenuated pathogenic vaccine, which actually isn't a good candidate for immunocompromised patients, transplant patients, the pregnant population. Mm-hmm. So it's a better, safer option, I think, actually.
Yeah, that's a good point. If you deliver it to where it's needed rather than kind of systemic circulation as well, people might feel less uncomfortable about it.
Right. And the reason for that is because this is a protein subunit vaccine, and it doesn't contain live attenuated pathogens that could be reactivated in an immunocompromised person. Mm-hmm.
Yeah, I guess Brittany said most of it, right? Like the key is really to make sure that people understand the process of how we do science and how we develop the vaccines, and that we're not starting anything without like a lot of testing, obviously, in humans and animals before. And as you said, Brittany, like I think like these new technologies that we're working on, like whether it's intranasal or like the particles are making, like they're bringing a new advantage also for the people, which is like the way it's delivered, which in a way might reinsure them. And that vaccine hesitancy can be driven by the delivery method and multiple injections, or a needle itself can like make people like fear a bit to get the vaccine. So having this new technology and explaining them how we develop them is I think the way forward to make sure that they will be confident into the vaccines.
Jenna?
Oh, sorry, I didn't realize that this question was also for me.
Oh, sorry. Yeah, we might as well, since you're working also on vaccines, how do you approach?
So I think Romain and Brittany answered this really well, and I think that just to perhaps add on to this is a little bit more on maybe what Brittany said is that some people have just accepted getting infected rather than getting vaccinated, but we know that infection is just not a safe option for a lot of different especially respiratory bugs. And related to some of my work is that when you get infected, it really changes the way that you respond, and not necessarily in a good way. Whereas when you get vaccinated, you can make it much more precise to the parts that you want to target. And I really try to stress that to people when they have questions about vaccines, whether or not they're safe, that you'll make a more focused and better immune response.
Yeah, that's a really good point, because the viruses and bacteria themselves have ways to subvert the immune system, and they can last and linger perhaps worse than the vaccines themselves. So contrasting what the alternative is to getting the vaccine might be a good strategy. Great. So our next question is for Guyon, and maybe also for other vaccines. So the rabies vaccine will be tested in other species like bovine and dogs. What is the process for how you move to humans to test safety and efficacy. What comes first in that process? What types of animals and studies do you have to do?
First, it will be tested in mice, and it's mainly to select the right formulation for the particles to deliver at the right time and mimic the prophylaxis, the post-exposure prophylaxis we're trying to copy with this technology. So mice is the way forward because it's easier to handle. It's easier to do all the preclinical experiments. Then we will move to another species for toxicity. For humans, so far, one of the major limitation is not necessarily the immunogenicity. It will be to make sure that we are able to produce enough particles to have batches that we can do post-production quality control on it. So the process, to answer the question, the process is first in mice, then some toxicity in a bigger animal model, which we haven't figured out yet so far, so I'm not going to say anything wrong. And on parallel, trying to increase the throughput of the platform is one of the key limiting factor to move to humans, mostly because of the fact that we need to validate the batch that we will make for humans. For animals, it's easier, the volumes are much smaller, but for humans, you need to have this kind of like engineering process in place before you start the trials. So that would be the way forward, I would say.
And is that similar for Brittany and Jenna for your approaches?
Yeah, so far we've done preclinical studies in mice. We actually do have some data from non-human primates, but the next step forward, we would be doing more toxicity studies in mice and additional studies in primates, as well as tox studies before trying to go for a clinical trial in humans.
Yeah, and I guess for our work, when we think about vaccines, of course, we use preclinical models, especially mice. But, the limitations with this when we think about epitope specificity of antibody responses is that animals and humans do not respond to viruses or, antigens the same, and we target different things. So really the motivation behind the organoid cultures is to provide kind of a preclinical human model to understand how humans could potentially be responding to vaccines before it actually goes into the clinic.
Great. And our next question is for Noam. Noam, have you tried to validate your programs to detect bacterial sequences with the tumors that have been associated with specific bacteria in emerging literature? Like I know there's kind of some emerging work on bacterial and viral presence in various tumors. Have you been able to check against that with what your programs are looking at?
Yeah, so we had a first validation was just based on the training data that we had, and the second one was in real tumors. We look at stomach and esophageal cancers, and we were able to see bacteria that are known to be there and are expected. Yeah.
Great. And are those specific to different tumor types and the different bacterial types? And what role do you think, what's going on there?
Yeah, there are different bacteria in different tissues and also in different tumors. The roles we don't know yet, but one of the goals is to see whether we see healthy tissue-specific bacteria and tumor-specific bacteria, for example, to see if there is maybe a cancer prevention role or cancer-driving role of those bacteria.
Great. Well, unfortunately, we're nearing the end of our session today. So I'd really like to thank our panelists for joining us. They'll be joining us in the networking lounge after the event. So if you have further questions, please feel free to join. You can click on the closing screen when we end. And there's also a link in the email you received to remind you of this event. The remote platform allows you to jump between small group conversations. So kind of as you would in a coffee break or a cocktail hour. So it's really fun. Come join us, give it a try. Also, I'd like to remind you this ePanel will be available on demand and we'll email you when the recording is available. I'd like to point out that we're having another Michelson Philanthropies and Science Prize awardee ePanel on April 11th. You can see the link in the event information tab on the left or in the reminder email for more details. So we hope to see you again soon for that. And finally, we will be hosting an in-person award ceremony for a subset of these awardees during our conference on B-cell biology in the context of infectious diseases, autoimmunity, and B-cell cancers, which will be held as a joint meeting with HIV vaccines, immunoprophylaxis, and drugs in Keystone, Colorado, June 6 to 10. For information on how to attend, see the link in the event information tab on the left or visit the Keystone Symposia conference page. If you register before April 6th, you can save $200 for that. So those are all my concluding announcements. Again, thank you all for joining us and for our audience for all those great questions. And we hope to continue these conversations in the networking lounge now. So we will see you there. Thanks, everyone.
Thanks so much.